Hi, hello, namaste. Welcome to Shrav's Biotech for You. We would now focus on the principles of biotechnology. There are two main principles of biotechnology that are 1. Genetic engineering, 2. Bioprocess engineering. Today's session we would focus on first principle, that is genetic engineering or manipulation. It is the technique used to change or modify DNA or RNA in order to obtain desired trait. It is the molecular genetic technique used for the direct manipulation, alteration, or modification of genome of an organism in order to manipulate the phenotypes. The term genetic engineering was coined by Jack Williamson in the year 1951. The types of the techniques under these genetic engineering are rDNA technology, gene delivering techniques, gene editing techniques. rDNA is nothing but the artificial DNA that is constructed by ligating two different DNAs using physical methods. Gene delivering techniques are the techniques used to insert gene of interest into the host genome. Gene editing techniques are the ones used to edit genome in which an undesired DNA sequence is removed or a new gene can be inserted into the host genome. Now moving on to the process of genetic engineering. The first step in the process of genetic engineering is selecting and isolating the candidate gene. Bacteria having a plasmid DNA is selected. The criteria for selecting the gene are: gene should have high GC content, low repetitive DNA sequence, and it should be smaller with a stop and start codon. This DNA or the plasmid DNA is isolated using restriction digestion or PCR technique. Second step would be construction of the plasmid. Plasmid must have ori, that is the origin of replication, promoter, antibiotic resistance gene. It is then inserted using restriction digestion or gene cloning. The plasmid DNA and the gene of interest are cut with the same restriction enzyme so that they are complementary to each other and they can be easily ligated to form rDNA or the recombinant DNA. This recombinant DNA is then transferred or transformed into the recipient or the host cell using gene delivery techniques such as electroporation, sonication, transposon or liposon mediated gene transfer, etc. Once this rDNA is now inserted into the recipient cell, this is then subjected to bacterial cell culture, where multiple copies of bacterial cells having this desired rDNA are formed. The recombinant plasmid DNA is then isolated from this bacterial cell culture in order to confirm the insertion of this rDNA. This confirmation is either done by inserting this recombinant plasmid DNA into microorganisms or through PCR technique. In case of microorganisms, the presence or absence of the selectable markers is used to differentiate between whether the cells have taken the recombinant DNA or they are still untransformed cells. With this, I wrap up today's session. Tomorrow's session, we would focus on the second principle, that is bioprocess engineering. Please like, share, and subscribe to my channel.